Okay, good morning, students. Thanks for coming here. We're glad to have our Masterclass. I'm going to give Jurgen Jones the opportunity to introduce his father as soon as we have a prayer. We're going to ask Logan to say that prayer if you would this morning. And then Jurgen will introduce, and we'll get right into it. Logan, will you be willing to pray for us?
Heavenly Father, thank you for this wonderful morning. Thank you that we all get to be here and take this excellent Masterclass with Dr. Jones. Thank you that we've all been able to make it here safely, even with the weather, and hope that the day will be great, and we'll be able to take into practice everything we learn and apply it in our lives, so that we produce Christ's name. Amen.
So, I could introduce my dad in multiple ways he probably wouldn't like. He doesn't really like any title he's been given him. So I'm going to give him my own title. My dad is a Renaissance man. He's a scientist, a poet, a singer, and a husband and a father. Please welcome Dr. Jones.
All right. We're going to begin with a little exercise. So look up there. Can we change the light a little bit for just that part? So you want to dim it, if possible. So this is a poem by James Merrill. Look closely at the letters. Can you see? Entering, stage right, then floating full, then heading off so soon. How, like a little coal-rimmed moon, O, plots her course from B to D, as Y, unanswered, knocks at the stage door. Looked at too long, words fail, phase out. Ask, now that body shines no longer, by what light you learn these lines, and what the B and D stood for. So what we're talking about today is how we know things. Some things we have to see. Some things we can't see until we've had other experiences and we can understand what we're seeing a little bit better. So I'm going to walk you through this again with a little bit of help, okay? Look closely at the letters. Can you see entering, stage right, then floating full, then heading off so soon? How, like a little coal-rimmed moon, O, plots her course from B to D, as Y, unanswered, knocks at the stage door. Looked at too long, words fail, phase out. Ask, now that body shines no longer, by what light you learn these lines and what the B and D stood for. So who among you completely understands the meaning of this poem? I bet you don't, Durgan. Anybody else? What does the B stand for? Very good. How does a body enter the stage of life? Birth. What does the D stand for? Death. And what kind of questions follow many people as they consider the afterlife? And by what light do you learn these lines? As bodies fail, these words, these letters sort of phase out, but as bodies fail, we die. There are many who would ask, why? There are good answers to those questions. Those questions will never be answered by science. What we're here to talk today about is science. And I do not mean science like science class, where you study biology or computer science, as I think all of you are studying this year, or chemistry or physics. Those are all the what of science. You are memorizing and applying things that other people have discovered. I'm talking about the how of science. Because science is knowledge in process. We make an observation, then we guess an explanation for that observation, and then we make a prediction that we can test with an experiment. Let me give you an example. People noticed that the earth was below, the sky was above, and both the sun and the moon seemed to be circling the earth. The guessed explanation was that the earth must be the center of the universe. The prediction would therefore be that everything should be going around the earth. Well, Galileo got his hands on one of the first telescopes, and as he gazed into the night sky, what he saw there was a planet, Jupiter. And he was able to see with his telescope, and this is actually quite amazing because it wasn't a very good telescope, but what he was able to see was that Jupiter had 4 moons, and they were traveling around it, not around the earth. And he used his telescope and Jupiter and its 4 moons to track its course, and found that it was traveling around the sun and not around the earth. So the prediction test failed. And the whole idea that the earth must be the center of the universe was rejected. Isaac Newton noticed that things fall to the ground. He explained this with the concept of gravity. The prediction from gravity would be that all things should fall to the earth. But, of course, not all things do fall to the earth. So did they discard the idea of gravity? Though sometimes, when a prediction test fails, we need to revise the prediction. So the gravity was revised to say it will pull everything to the ground unless there is an equal and opposite force in the other direction. This led us to learn something new and ask new questions. We started paying more attention to the bird and the bird's wings and think of all the ideas that have flown from that line of thinking. So it is the test failures, the exceptions, the outliers, that teach us what we don't actually know and lead us to new questions. Failure, or the opportunity or potential for failure in this prediction test, is critical. So Sigmund Freud styled himself a scientist, but what he did wasn't really science because he could not fail. So he would explain almost any behavior that a person exhibited as being due to the fact that if he were a man, he was in love romantically with his mother and hated his father. And so his prediction was that every man hated his father and loved his mother. But he would only test this by asking people. And if you responded to him, oh yeah, you're right, I totally hate my father and am romantically in love with my mother, he would say, bingo, you're right. Proves my theory correct. But if you said, I don't really think I hate my father and am romantically in love with my mother, he would say, you're lying, I'm still right. So his prediction test could not fail. So it wasn't science. At least wasn't the scientific method. Failure is critical. An explanation that cannot fail is dogma. Dogma is just knowledge that's based on authority. You should believe this because I'm telling you this, not because I'm showing you evidence or I've proven it even to myself in any way, just because I'm saying it. This is frequently used by our favorite scientist celebrity, Bill Nye, the science guy. He's always saying things like this. Well, 97% of scientists agree with this or that or the other. That's not science. That is using authority of scientists as, I don't know, I guess we think smart people in our society. They aren't necessarily, they may be. But it's using their authority. It's not using this process of science, of testing predictions and making predictions. And it's also why he was always trying to sort of grasp at these elements of authority. This is an actual quote from him. I took 6 semesters of calculus. Is that enough? Is that enough? Partly I say, most people get through calculus in 3 semesters, so I'm not really sure why it took you 6. But more importantly, calculus is not the practice or method of science. Because calculus is already accepted principles. The calculus that you all will learn here at AHS is the same calculus that I learned, the same calculus your grandparents learned, and the same calculus that your grandchildren's grandchildren will learn, because those principles aren't changing. It's not that there was never maybe science involved in calculus in its development, but the learning of it is just memorizing somebody else's principles and applying them. It's the same with engineering. Again, this is not to say that these things are bad, it's just that they are not exactly the scientific method. In engineering, we apply best principles. The only time when engineering becomes science is when those principles that are predicted to work fail. This is the Tacoma Narrows Bridge, a suspension bridge that was built up near Tacoma, Washington. And they applied the very best engineering principles of the day. That was not science. It became science when they realized in practice that the winds going through the Tacoma Narrows were too forceful for the structure to withstand, and the structure failed. Failure is critical in science. So that was a moment where engineering became a science experiment. Also, forward is critical. Remember, this is a prediction. That means saying it beforehand. You can't say it beforehand if you're testing something in the past. The most frequent thing that irritates me as people talk about science is they talk about Darwin's theory of evolution and his book, The Origin of Species. What Darwin did in that book is very careful observation of a number of the animals that they found in the Galapagos Islands. And he found specifically among the finches that they had different kinds of beaks on different islands in the archipelago. That is an observation. The guest explanation was that those beaks were selected for by natural selection because of the availability of different food on the different islands. The prediction was therefore that all finches began as one finch species, and then it drifted from there in different ways. How do you test that hypothesis? How do you test that prediction? You can't. You can't go backward and test a prediction. It's a prediction. We have to say it beforehand. Which is why whenever anybody says from this, well, this proves that scientific evidence shows that man came from apes, I would say, it does not. As a scientist, I would say, it does not. It's plausible that this is one explanation. There are many other plausible explanations, but none of them can we test, because all of those are retrospection. A retrospective explanation may be believed, and you may believe it on good authority. You may believe the people who came up with it. They may sound more trustworthy to you. But only its feasibility in the forward-going sense can be tested as a prediction test. So natural selection, the process of any species reacting to its environment by selecting for the most fit individuals within that species, that is a principle that you can apply in a petri dish and you can watch it happen. If you apply some chemical selection on bacteria, you will select for the few bacteria among that whole population that are already resistant to that chemical. And they will grow more. And so more of that population at the end of this selection pressure will look like that original small part of the population now expanded. Natural selection as a principle, which is what Darwin was trying to get after, is a very true principle. You can make it happen in front of you. But you can never prove that that is where any species came from, because you can't look backward. That's why one of my very favorite phrases to hate is science is real. I see this on billboards, I see it on signs around my neighborhood. I have no idea what that means. I'm a scientist, I have no idea what that means. Most of my science is biology. And as you all know from Dr. Matt, Mr. Matt, he's not here, from his class last year, biology is very variable. There are many forms of life. And so everything we study in biology is about populations, meaning that everything is distributed around a normal curve or a Gaussian distribution. And so our idea of an observation is usually the middle of that curve, of that distribution of a population. And if we are trying to add an arrow onto the map of biology, of the factors that affect this and that and the other, we're going to test it in populations. And ideally, we're going to test it with a prediction test that only changes one variable between two populations that are otherwise the same, and then to see, do they differ in the outcome of another variable? And if they do, and if that difference between their means or their averages is statistically significant, we might accept that arrow as real. But important to our paying attention to this is the fact that there's a lot of overlap in between. There are very well might be individuals in each distribution that are past the mean of the other one. It is also true that any knowledge we have from a population, or any biological knowledge that we think we have, is only true as far as the next outlier. That, like Jupiter's moons, is going to teach us what we didn't actually know. Our principle wasn't really perfect. But those outliers can also teach us something else. They're like a tree standing outside the forest. Now, we always say, don't lose the forest for the trees. But you also lose a tree within the forest. If what you're trying to understand is what is the relationship between trunk and roots and branches that make a tree, get a tree out by itself, even if it's a crooked tree, even if it has an unusual relationship between trunk and roots and branches, even if it is growing in an incredibly unusual circumstance for a tree, it nonetheless draws our attention and allows us to do that first step of the scientific method, which is to make an observation. So most of the science that I do is actually focused on cancer. And cancer is always an outlier when it comes to biology. It is biology gone awry. Sometimes we are studying cancer to try to kill cancer. Sometimes we're studying cancer to try to understand the general biology of cells and how they work. Most of my science is focused on a type of cancer called sarcoma. Sarcoma is absolutely an outlier among all cancers. Sometimes we're studying sarcoma to try to figure out how to kill sarcomas, because we're trying to kill our patients. Sometimes we're trying to study sarcoma because we're trying to understand the general rules of cancer biology. So sarcomas are rare. They make up about 1% of cancers. You probably also know that cancer is considered a genetic disease. We know that because there are tumor suppressor genes that have to be turned off in order to form a cancer. We learned about tumor suppressor genes even generally, even though they are important for almost all cancers. We learned about them from these itty-bitty tiny fraction of cancers called sarcoma, because Drs. Lee and Fermani noticed that there were some families that had way too many sarcomas. I told you sarcomas are rare. If you've got a 1 in a million diagnosis and you've got a brother and a sister that both have it, that draws your attention and it makes you say, hey, something else is going on here. That's what Drs. Lee and Fermani did. These families also had more breast cancer and more all sorts of other kinds of cancer, but they noticed it because of the sarcomas. The exceptions, the outliers, really draw our attention and allow us to make that observation. However, at first it was only an observation. So they still hadn't proven anything. The observation was that TP53, this gene, was lost in many cancers. The guess explanation was that TP53 loss causes cancer. The prediction was that if you turned off the gene TP53 in an individual, that it would cause cancer. But remember, this is a prediction. So we can't go back. Any time I see a patient that comes in, even if I fully sequence the whole genome of their cancer, and I see a TP53 mutation, I still don't know if that has actually caused their cancer. Because no matter how many times we observe something, that is not a prediction test. And it's very unlikely that you're going to convince people to say, how about if I take you and your brother, and I'm going to turn off your P53, and I will turn off his? Most people would not sign up for that. So we started making knockout mice. This is where we can manipulate the genes of a mouse. And we can see, since a mouse and a human have very similar genomes, we can see if we turn off a gene in the mouse, can it make this disease that is associated with that disease in humans? So the guess explanation again was that TP53 causes cancer, and the prediction was therefore that silencing mouse TRP53, has a slightly different name, induces cancer, and indeed it does. In medicine, we so often think that it is kind of an engineering problem, that it's all solved. So we have the boardrooms of insurance companies and the hallways of Congress that are trying to figure out how to pay for it, ethicists and epidemiologists trying to figure out how to distribute medicine, and physicians in hospitals are really just totally consumed with the idea of trying to practice medicine safely. These are all noble efforts, but they also assume that the textbook is closed. And in this era where we start to measure quality in healthcare by how quickly we can get to it, and many of our provisions for healthcare, our institutions for the provision of healthcare, starts to look a heck of a lot like Jiffy Lube. The problem is that I don't have one of those little doohickeys to plug into your car and find out exactly what's wrong with you. Because the textbook of medicine is not finished. Medicine is science, it is knowledge and process. We make an observation, we guess an explanation of that observation, and then we make a prediction that we test with an experiment. It was not always that way. Back in the day, medicine very much tried to be dogmatic. This is actually a textbook from the Middle Ages, as they have this guy, looks like he's on a rack. They're actually trying to straighten out his tibia, which is healed in a crooked position. Medicine in the Middle Ages was primarily the purview of the Catholic Church in Europe. And so there are often woodcuts depicting things like this, where you lay out the instruments, have a prayer together, and then cut off the limb. I don't know about you, but if somebody had my leg braced across two boards and was getting ready to jump on it, to break it, to straighten it out, I don't think that I would have this pious little smile on my face. But this was an era when medicine was all about authority. It was not about asking questions. It was not about trying to prove anything. It was about authority. Samuel de Sorbier was a physician writing advice to young physicians. This is in 1672, so quite a bit after some of those pictures. But even then, he says satirically, I must tell you that medicine is a very imperfect science. It is quite full of guesswork that it scarcely understands its subject matter, nor is it familiar with the things employed to maintain it, that the more enlightened only feel their way in it, groping amidst a thick gloom. Could not have said a truer statement. This is the era when bloodletting and leeches were considered a great idea. What he thought a physician should do is bestow a few doses of nonsense. And his reasoning is fascinating. If you don't tell them what they want to hear, they will go someplace else. They will reject your authority as a medicine. More than 100 years after that, this is a Lancet, so still one of our very prominent medical journals today. But this is an editorial. Much also depends on the steadiness and mild, though confident manner in which the surgeon himself sets about his painful duty, inspiring his victim with reliance on a certainty of relief at the expense of a temporary amount of suffering. Surgery in 1836 was only amputation. This was before anesthesia. This was before the idea of the germ theory. You were good if you were fast. So when they were amputating a limb, like an arm or a leg, they would typically, with a very sharp blade, wrap your arm around the limb you're going to take off, and then with a slash of the blade, cut down to the bone, grab a saw from an assistant and saw through the bone to try to get the limb off before the patient fainted or died from blood loss and everything else. You were good if you were fast. So Sir Ian Liston, he was so fast he was knighted, is also famous in medicine, or infamous, for being the only person I know of to have a 300% mortality from one surgery. In the flash of the blade, he accidentally nicked the chin of the family member standing a little too close, and the finger of his assistant who was holding the limb for him. The patient, the assistant, and the family member all died within a week of gangrene. But they tried to have authority. Many of you might recognize St. Peter's Basilica in Rome, a religious symbol for many around the Christian world. You probably don't recognize this hospital, which is built in Baltimore in 1890, a long time after the days when churches were primarily building hospitals, and many hospitals looked like churches. Why did they build it to look like a cathedral? This is Johns Hopkins Hospital. And inside that dome, you will recognize this statue from Temple Square, the Christus statue by Earl Thorvaldsen. A replica of that same statue stands right in the middle of that dome of Johns Hopkins Hospital. It looks as if the whole hospital was built around it. Until you read your history book and you realize that that statue was added 2 or 3 years later as a PR campaign because they worried that they weren't getting patients to trust them. The symbol of medicine throughout most of the world is the Red Cross. That is not an accidental reference to Christianity. And in fact, in much of the Islamic world, it is a crescent instead of a cross. This is medicine trying to grasp at authority instead of evidence. It is putting dogma in between a patient and the physician. All this began to change about 20 years after that hospital was built by one man named Ernest Amory Codman, who suggested that maybe we should measure an intervention in medicine, if I'm doing something to a patient, by what it actually accomplishes for them. Maybe we should look at their outcomes. How do we help them? So we started calling patients back like 6 months after they had surgery. And it didn't look that good. And in fact, it was so bad that Harvard, Mass General Hospital, that was his employer, fired him for daring to suggest that we should actually call patients back and look at their outcomes. Furious about being fired, he put this cartoon in the Boston Globe. And what's even more fascinating to me than the fact that he was fired by Harvard for trying to bring science, or at least honest science, to medicine, was the response of the public. The public didn't see this and say, oh my gosh, like, this is so right. We should be measuring this carefully. They said, how dare you suggest that we don't know quality when we see it? And they ran him out of town. He died a pauper. He did usher in this idea that maybe we should start measuring results in medicine. And about 30 years later, even Harvard repented and started doing things scientifically in terms of treating patients. But the challenge is that every time anyone goes to a doctor, they're going to be a part of an experiment. We talked about how an observation is in the median or the mean, the middle of the population. But it's a prediction test for every individual whether they're going to be on either side of that mean. And that means that they're either going to do better or worse. So when we look at it from a distance, medicine looks so clear and confident and so full of authority. But when we look at it closer, there are some splotches and some broken bits and some bandaged up places and even the occasional black eye. The question is, how are we going to face it? How are we going to tolerate these asking of questions, which we're going to experience? So do this little exercise with me. What is written up there? Okay. What if I move this space? How do those two relate? Like complete opposites. All we did was shift the space. So maybe it matters a lot where the space is. In science, what we're always trying to do is pay attention to the gap in knowledge. What do we not know? And that might be something that we don't know for an individual. Someone coming to medicine to have a procedure. And do I know whether it's going to work or not? I'm not sure. I'm going to work with you to try to do the best we can, but you need to understand going in that I cannot perfectly predict it. As I spend all my time with patients, and I spend my time doing science, I find that the most important characteristics are humility, which basically means a willingness to accept when I haven't known as much as I thought I knew. And curiosity, meaning I'm still seeking for those moments where I didn't know as much as I thought I knew. But also, a big part of every experiment that we do is faith. Every prediction test requires that we have some belief that, number one, I'm going to get an answer. Number two, that I believe this principle if I'm going to apply it. Now, since this is a Masterclass and this is supposed to be kind of like seminary, how does this fit with the gospel of Jesus Christ? So, we are not practicing dogma. Many religions do. Our religion is a religion of questions. It is where individuals are invited to come into Christ and ask their questions. It began with a young man asking a question. We even have experiments. In Alma, it talks about, but behold, if you will awake and arouse your faculties, even to an experiment upon my words, an exercise of particle of faith. Yet even if you can no more than desire to believe, let this desire work in you, even until you believe in a manner that you can give place for a portion of my words. And giving place for a portion of his words is basically planting a seed and seeing what happens to that seed. Will it grow within you? We test God's principles. Importantly, we do not test him. That doesn't go well. There are a few examples of that in scriptures. It doesn't go well. But we do test his principles. He says, prove me now herewith, and he's talking about tithing. Prove me. I'm going to make a prediction that tithe payers are blessed. The guest explanation that paying tithing enables God to bless me. And the prediction is that if I pay tithing, I will receive blessings. Once I've done that and experienced that, then it's not the same as belief. Then I start to know something. I might forget, and I might have to be convinced again by another prediction test. But then I start to know. We also test ourselves with respect to his principles. And this is the main way that we do prediction tests in the gospel of Jesus Christ. In Joshua chapter 3, this is when they're about to cross the River Jordan to attack Jericho, actually, and the Lord has said, I'm going to give the city of Jericho into your hands. But he says, as they come to the River Jordan, and thou shalt command the priests that bear the ark of the covenant, saying, when ye are come to the brink of the water of Jordan, ye shall stand still in Jordan. And it shall come to pass, as soon as the soles of the feet of the priests that bear the ark of the Lord of all the earth, shall rest in the waters of Jordan, that the waters of Jordan shall be cut off from the waters that come down from above, and they shall stand upon an heap. How far into the water do you have to go? how far do you have to test that prediction in yourself sometimes? One of my favorite stories in the Old Testament is the story of Shadrach, Meshach, and Abednego. So these are young people growing up as basically captives, taken away from Jerusalem, growing up in Babylon. And at one point, and there are a bunch of these are all in the book of Daniel, a bunch of these stories where basically ultimatums are given to Daniel and his three friends, Shadrach, Meshach, and Abednego. And this one is particularly that they are commanded to worship a golden image of the king Nebuchadnezzar. And this is their answer to the king. If it be so, our God whom we serve is able to deliver us from the burning fiery furnace, and he will deliver us out of thine hand, O king. But if not, be it known unto thee, O king, that we will not serve thy gods, nor worship the golden image which thou hast set up. So they're saying, the prediction test on me, I'm going to do it no matter how it turns out. And often that's our task. So I have told you about asking questions for a living, which I do as a scientist, and asking questions for a life, which is what I try to do as a disciple of Jesus Christ. So back to that poem in the beginning. Ask, now that body shines no longer, by what light you learn these lines, and what the B and D stood for. I think the key to asking questions is humility, curiosity, and faith. Because failure is not only a critical part of science, it is a critical part of life. Everything we do is a bit of an experiment. Everything we do is trying to test what we know and what we think and what we believe to try to understand if it's going to come true for us in the next day. I say these things in the name of Jesus Christ. Amen. Amen. Any questions? We've got a few minutes. Okay. Yes?
So earlier when you were talking about, I think it was the gene that...
TP53?
Yes.
Yeah.
I was just curious, sorry, I'm bad with words sometimes.
It is early in the morning. It's all right.
Do you think it's possible that there is a gene like that causes other genetic diseases?
Absolutely. I mean, that is one sort of classic big example, but there are many genes. There are, about 20,000 genes in the human genome, and not all of them, but many of them, if turned off, will be associated with some disease. Not all of them cancers, obviously. There are lots of other genetic diseases. Yes? That's a good question. So usually TP53 is turned off by specific mutations. So the sequence, if you remember that from Mr. Matt's class, where you have a DNA sequence and then that gets transcribed into messenger RNA, and then that gets translated into a peptide sequence for a protein. There are many mutations in TP53 specifically that will change one amino acid or change the code from one amino acid into a stop codon in the middle of a gene, which means that you end up forming too short of a gene or just no gene at all, no protein at all. And so how those develop, I often explain this by, if you think back to the Middle Ages, when the monks were copying the Bible, they'd have one copy that they're reading from right here and one copy that they're writing on right here, and they're copying the letter at a time. They're going to make some mistakes, and a lot of those mistakes will mean nothing. But what if they leave out a no or a not? What if the wind turns into wine? Like, it could have a totally different meaning for one letter changing. And so the thought is that there is a copy of your whole genome in every cell in your body. And there is a certain rate of making mistakes as that is copied each time. A few trillion cells in the human body. Even if you have a very low rate of mutation, you're still going to form some. Most of those are going to happen in the sort of non-critical words. But every once in a while, someone's going to change the meaning of the sentence, maybe even the whole paragraph, maybe even the whole chapter. And those kinds of changes are what's going to lead to diseases or cancer or something like that. Yeah. Yes?
So you gave us a definition of science, essentially, and this process of asking questions and testing with predictions. You seem to have embodied this in your life. What specific invitations or challenges might you issue to these 40 young people to double the amount of science that they're doing every single day, every single hour?
So you're doing it. Whether you realize it or not, what I would say is sort of a healthy way of asking questions is to do it consciously. If you remember from the poem at the beginning, when you first look at it, you're like, oh. But as you start to pay more attention, it starts to mean something different. And I can guarantee you can go back and read that poem on your own, and you'll probably get additional insights. It's a simple, silly little poem. But when we pay attention, when we are aware of the questions that we are asking with our lives, with the principles we're applying in our lives and trying to see if they work for us or not, if we do that consciously and honestly, I think it's healthier. I think it's happier. But you're all asking questions, whether you know it or not, because you don't know how the day is going to happen. You're not sure that the sun's going to rise tomorrow. You predict it will. I predict it will too. But it's going to unfold as we watch it. So paying attention to what we're observing in our surroundings and what we're experiencing and what principles it's confirming to us. Yeah. Other questions?
You've written a book or two.
Only one, yeah, not two. Yeah, I've written a lot of scientific articles that you would not want to read. They're quite boring. And I wrote a book about uncertainty in medicine. Because I like stories. And I don't think there's anything more dramatic than teenagers with cancer. And that's what I treat mostly. So if you are a 2-year-old and you have leukemia, it's a dramatic experience for your parents, probably for your grandparents. I don't know that you are even that aware of it. If you're a 14-year-old, if you're a 15-year-old, 16-year-old, and you have a cancer that is a devastating problem in your knee or your thigh, not only is that totally affecting a person who understands what's going on a little bit, but they also have to make decisions. And so the kind of cancers that I treat, I'm working with teenagers making decisions about what they want to do with their limb and the tumor that's in their limb, how they want to reconstruct it, what kind of an adult they plan to be. And those kinds of experiences are very intimate and very, very sacred. But it's amazing to get to participate in those with patients. That's why I do what I do. Yeah.
So back to the degree, is it possible to turn it back on after?
Good question. A lot of people are trying. People have been trying for about 50 years. Hard. hard to undo a mutation, unfortunately. Yeah.
Can you tell us a story from experience that you have with a patient?
We're probably on low on time. So, a quick story. So, friend of mine who is another surgeon, he's been gone for years now. He saw a patient with a tumor in her foot, and he took this tumor out, and his conversation with her was, "I got it, you're good to go. You know, she's fine." She and her husband, celebrated a fancy dinner out. In our meetings, he said, "I'm really worried about this one. This is a type that really comes back frequently," and we all said, "Yeah, it is." When it did come back, when it turned out he hadn't gotten it all and she wasn't good to go, he asked me to see her instead. Like, you know the right thing to do as much as I do. You haven't done anything wrong. And he's like, I can't. So I met with her and I had a very different kind of conversation. But the critical thing was, I said, this is what we're going to do. It's an experiment. We may or may not eradicate this tumor, and this is how we're going to watch for it, and we're going to work together. We're going to be a team now. Less authority, more of a teacher, counselor. And she actually just finished her 10-year surveillance about a year ago, and she's very happy. She's been okay the whole time. But all the way along the way, we were acknowledging that there was uncertainty and that we weren't sure how it was going to turn out. And I think that's really critical. So, okay, thanks everybody.
Okay, and I think it's someone's birthday today. Evie's birthday. Is Evie here? And Durgan's birthday. And it's also Matt Shirt's birthday too, right? There are probably some other students in the school. All right. Well, Mrs. Jones brought some treats for all of you. It's a cookie or something like that in the bag. It's a Ziploc bag, so keep it for lunch and then give Durgan a big hug and a kiss. No, I'm just kidding. Give a kiss. At lunch. Thank you so much, Mrs. Dr. Joyce. Let's give her more round of applause. That's all I ask. Lots of questions and be conscious about the experiments and the predictions we're making. Be reflective to it in your journals. Have a wonderful day. Take care. Let's see. How many of you are coming to the play tonight or tomorrow? Okay, we hope you will. Invite your families. It's going to be great. 7 p.m. tonight, 2 p.m. tomorrow, 7 p.m. also tomorrow. Next week, we have Mr. Russ Schertz, who's a lawyer, and that's Savannah's dad. So Savannah, will you prepare a brief introduction for your father? He's going to share some of his most important mental models. We hope you all come. Please be on time too, so just 8:15 we can get started. One last announcement is that January, if you didn't see this message, in January, we're actually having the 10th graders and 11th graders start with Mr. Murdoch's class at 8:15. And I know seminary, sometimes you might say, well, there's not a lot of homework in seminary, but unfortunately seminary has to change their schedule and their human resources, so we've got to flip our schedule. So you 10th and 11th graders need to be here at 8:15, so you are here for British literature with Dr. Murdoch. Okay, right, have a great day. Let's go to the next classes. Bye-bye.